So my apologies. I was trying to get the multimedia up and running for today. Clickers, most people have registered. I've still got 19 of you who think they're registered for the class and don't have their clicker numbers. Yeah, that's only one of you, unless you're like a clone of 19 people. Got 19 clickers in the back? But some of the people with clickers who don't actually have scores yet, those clickers have like 16 points. So I would highly recommend that you either guess a clicker number, maybe, if you want some more points, or send me your Odin username and clicker number so that we can connect the two of you. So that's there. I tried to upload. The problem uploading takes about 20 minutes, and so it takes a little while for all of that to go through. After class today, I'll be uploading those scores. So any questions about clickers and so on and so forth at this point? Yes, we're all happy. We all have them. They're ready to go. Good. So let's get started with our clicker questions for today. And they are, if I could actually start the clicker software, that would help. See, I knew there was something I was missing here. Let's do this. You see, there wasn't my little cute little sign thing on here. So obviously, it was not working properly. Searching for bases, searching for bases. There we go. Does that look better? Yes? Good. So now, measles is a paramyxovirus, rhabdovirus, filovirus, picoRNA virus, or flavivirus. I thought it was supposed to count down rather than count up. Oh, well. One of these days I'll figure out the technology. 5, 0. It's not stopped. Oh, no. Okay, paramyxo, rhabdo, philo, picoRNA, flavies. No, it's not. We're going to be talking about today our friend Ebola Zaire. That is a filovirus. So what did we talk about last time? Paramyxo and rhabdo. I always try not to have something which is exactly on any of the slides, so you can't go back and do your search term and try and find it. But yes, it's a paramyxovirus. Rhabdoviruses are what? Rabies and some other ones as well. What else? VSV, vesicular stomatitis virus, as well. So our next question, receptor binding protein VSV is called HANG-GP30HN. And you're all doing your revisions for the midterm, right? So you all know this. Part of the reason for these questions is they're quite similar to what will be on an exam. I'm obviously giving you far too much time. So the receptor binding protein in VSV is G. What's HA? Hemagglutinin from what? Influenza virus. N is what? Nucleocapsid protein. G is the receptor. GP30, we'll talk about today. HN, receptor binding protein for. What was the answer to the last question? Paramyxoviruses and measles. Yes. So most of you have it right, but I thought I'd go through the process here. So, okay. Adios, amigos, amigis. Today, we're going to talk about filoviruses. I keep forgetting to rename this lecture because it says philo and bunyaviruses. We're not going to talk about bunyaviruses. I wish we were going to. I may be able to convince Dr. Uedas from the biology department to come and talk about his favorite bunyaviruses, which are the Hantan viruses, at the end of the term. We'll see if I can be able to convince him for that. But the main reason, of course, for filoviruses is our friends currently in West Africa that we'll cover towards the end here. Molecularly speaking, filoviruses are actually kind of boring. They are basically these big paramyxo/rhabdoviruses, and when we look at their genomes, we'll see they're actually really quite similar between those two. There are some very interesting differences, which may be why they cause these really nasty diseases, at least in humans. As far as we can tell, that's actually not the normal reservoir species. This is true for many of these viruses that cause nasty diseases. So, main things here, okay, they're basically rhabdo or paramyxoviruses, at least at a molecular level, with a couple of differences. One of them is RNA editing. They actually have a very specific editing function that happens in the replication of the viruses, which does seem to be absolutely critical for disease and replication. And then, of course, this, what did we say last time? Gross disease phenotype, something like that. Although what's interesting is the more you look at the current outbreak, actually there's not that much of the hemorrhagic cases which are happening. It's much more of a general, nasty disease, yes, but there's not that much of the hemorrhagic aspects of it. So we'll talk about structured genome protein editing regulation and then flip things around a little bit. We'll do the origin and disease right at the end here. This, unfortunately, should be a dot. This is probably one of the most famous EMs around these days, taken by Fred Murphy in the late 1980s. So people have known about Ebola and these filoviruses for a really long time. Unfortunately, what's not on here is a scale bar. These virions are enormous. They're really, really big. They're almost a micron in length, which is much, much, much bigger than anything else we've talked about so far. They're almost visible under the light microscope. These are really, really large virions. Not quite as big as the giant viruses we'll talk about later, but they're really big. They have, again, a lot like, and you'll probably hear me say this many, many times today, the paramyxo and rhabdoviruses, this genome in the middle, which is packaged with a nucleocapsid protein. I'm not sure this interesting typo here, nucleoprotein. And then one of the big differences here is this VP30 protein. And we'll talk much more about VP30 as we move on through the rest of the lecture today, particularly thinking about some of the vaccines. Just recently, I think about 2 weeks ago, there was a nice publication in Science about a vaccine that's lacking this VP30 protein. So the VP30 protein is absolutely critical for replication. So again, a big difference there. Otherwise, we've got a nice membrane. These are enveloped viruses. There's a matrix protein. There's a glycoprotein. This glycoprotein is serving extremely like G, which is kind of the reason that I had that as the clicker question for today. This serves as both the receptor binding and fusion protein for these guys. Negative strand RNA viruses, so they have to have a polymerase over here at one end together with other proteins, which are not that critical. But again, it's bound to the genome, so when it comes inside the cell, you're immediately going to get some kind of messenger RNA production from that opposite strand. Remember, this is going to be now bound at the 3' end of the genome. What does that genome look like? Again, very similar to paramyxo and rhabdoviruses, which in fact I have down here at the bottom. This is our classic paramyxovirus. We've got N protein, this funky PCV protein, and then finally down here at the end, the L protein, gene, I should say. Why are they all organized like this? Relative concentrations. You need a lot more of N than you do of L. How does that happen? Starting and stopping. So the RNA-dependent RNA polymerase comes along here, gets to one of these intergenic sequences, copies a few of these U residues into A's. That's the stuttering. Probably most of the time we'll dissociate. Some of the times we'll get back, not even get back on, stay on the template, and then make the next messenger RNA, etc. Very, very similar things happen with these genomes. One thing is that this one's considerably bigger. It's 19 kb, so it's a pretty darn big RNA genome. The RNA-dependent RNA polymerase here, unlike all of the RNA viruses with the exception of the coronaviruses, is what? Low fidelity, makes lots of mistakes. And so there was a lot of discussion, particularly in this outbreak, about massive numbers of mutations happening in the genome. Well, it wasn't just massive numbers of mutations happening in the genome because of the outbreak. It was massive numbers of mutations in the genome because it's replicating and it doesn't have proofreading. So makes lots of mistakes as it goes to the genome. And again, there are just a couple papers that came out recently that showed that the replication error rate is no different in the current outbreak than in any of the previous ones. So it's not evolving faster, as many people have said. So nucleocapsid protein, NP, by far and away the most of this. And then each of these start-stop here as well. What's a little different between the filoviruses and the paramyxos and the rhabdos is that in some places you actually overlap the start and the stop. So here, this would be your standard set of poly-U's that, just like down here, would get copied into a poly-A tail. Only instead of the polymerase staying on and keeping going here, it seems to back up a little bit. The actual place where the transcription, I should say the messenger RNA production starts, is right in here. So there are a couple of places like that in these filovirus genomes. Caps are put onto all of these messenger RNAs. Again, where's that coming from? Viral proteins, particularly this L protein. Again, just like the case in the paramyxos and the rhabdoviruses. Proteins, again, ridiculously similar. Start out here with the GP, the glycoprotein. It's a little confusing, a YG for the receptor binding and fusion protein. It's because these proteins in both the rhabdoviruses, which is G is from, and these filoviruses are really heavily glycosylated. They've got lots of sugar residues hanging off the outside of them. And that's probably to help them escape the immune system. And that seems to be the reason, and that's why a lot of these things are called G. Because they're heavily glycosylated. When you purify virions, you find one protein that's highly glycosylated. That's the one which is sticking on the outside. Seems to be, again, important for receptor binding and fusion. So we could, take, let's see, the G protein of the rhabdoviruses, HN and F of paramyxoviruses, etc. So this, being on the outside of the virion, it's course, what's dealing with the immune system. So this is the basis for the vaccines that are currently in trials in West Africa right now. There are, again, other ways that people are looking at this as well. Put the box down here around the ones which are different, VP30 and VP24. VP30 is really important for making messenger RNAs and making the proper messenger RNAs. And we'll take a look at that in just a couple of slides here. And again, very different between the phylos and the rhabdos and paramyxos. There's this VP24 as well that is also not present in the paramyxos and rhabdos, and we really are not quite sure what exactly it's doing here. The other ones, again, are extremely similar as far as how they're functioning. GP is, again, a lot like the G protein that we find in the rhabdoviruses. It's made as a precursor protein. Again, one big polypeptide that then gets chopped. That chopping process seems to be critical for exposing the fusion peptide. And again, this is a really common theme that we see again and again and again, and we're going to keep seeing throughout the rest of the course. It's cleaved by a cellular protease, so there doesn't seem to be an obvious viral protease encoded in the genome. It's all cellular proteases. And again, highly glycosylated, made in the ER and the Golgi. It's a trimer, et cetera. There's one slight difference here with this GP protein in particularly Ebola viruses, and we'll talk about Marburg as well a little bit later on. It's what's called the secreted glycoprotein. And so when you're infected by one of these viruses, there's also a lot of this separate protein here that's not attached to the membrane, floating around inside, usually, unfortunately, the blood of an infected patient, which may have to do with some of the nasty disease phenotypes. We don't actually know what that is. One theory is that, again, because it's all those sugar residues that are helping you hide out from the immune system, but also what the immune system recognizes, maybe what happens is the immune system recognizes all of these glycoproteins that are not associated with the virion, and it sort of sucks up all of the immune system proteins and macrophages, all those good cells, where then the virion can actually survive under those kinds of conditions. But again, this is purely hypothetical at this point. One idea that people have is to try and get rid of this secreted glycoprotein and see if that might work also as a vaccine. That hasn't progressed as far as some of the other ones. And again, what it's binding to is still a little bit of an open question. Seems to vary a little bit from virus to virus. Transcription, replication, and assembly. Again, sorry to keep beating the dead horse here. But the main thing is that start, stop, stuttering, make an antigenome, make a genome. Again, all of this is similar to what happens in paramyxos and rhabdos. So it's a nice review. How do paramyxos and rhabdos know when to make genome and antigenome? Concentration of N, which in the filoviruses is nice and confusingly named, NP. But N and NP are usually pretty interchangeable. Buds again at the plasma membrane. Probably GP40 is important for this, which is, again, matrix proteins sitting in that membrane and sort of seems to be providing an interaction between your nucleocapsid and the outside. Difference between filoviruses and rhabdoviruses has to do with how this secreted and non-secreted glycoprotein gets made. The insertion point, maybe I should back up really quickly here, for this protein is at the C-terminus. So at the C-terminus, you've got a nice amphipathic, and in this case, completely hydrophobic side chains that inserts into the membrane. That's encoded for by this brown open reading frame right here. What's curious is when the genome was sequenced, what they found was only the sequence for this secreted glycoprotein, which doesn't have this C-terminal end with that hydrophobic tail in it. People went back, they looked at the messenger RNAs that were being produced, and it turns out that those messenger RNAs that are being produced have an extra residue, which has been added into that messenger RNA. So it's not a change in the genome. It's just an addition in the messenger RNA. Now, what is sort of a smoking gun as far as that's concerned? Editing of messenger RNA, changing open reading frames. What's it coming right after? Whole stretch of lysines, yes, but what are these lysines coded for? Poly-A tract. So, how are we getting this editing? Stuttering. So it's very, very similar to what you're getting with the poly-U's. So, in this case, about 80% of the time, get your normal, non-hydrophobic tail. 20% of the time, add an A, and you change this. How do we know this? People have done mutagenesis of the genome, where you take each of these lysines here and just change the codons. One of the great things about the genetic code, it's degenerate. So you can make lysine here. So the protein's exactly the same, but your codons are different. If you do this, then you end up with 100% of this secreted glycoprotein, none of the membrane-bound glycoprotein. Now, how do you do these kinds of experiments? Where are you going to be working with a virus like this? Any ideas? In my lab here at PSU? You want me doing these experiments? No. So these are all going to be done in high-level biosecurity labs. If you're working with Ebola or Marburg fever, that has to be at BSL-4. There are a few of these Ebola viruses, like the Reston Ebola virus, which actually are much less pathogenic to humans, so they're a lot easier to work with. One place is Hamilton, Montana, just outside, about an hour south of Missoula. Another place is Galveston, Texas, which is with a group that I collaborate with, although they don't work with this. And then there's another BSL-4 lab, not surprisingly, at the CDC in Atlanta. They're trying to get one in Boston. It's a whole different story that we may have a chance to talk about a little bit later on. But if you want to do these things and do these kinds of experiments, you do need to have these high-security labs. So that's how you get secreted versus non-secreted. And again, the idea here for the vaccine is if somehow you could flip this around. You could now have only this protein and not make this protein, whether that would be any better. Still an open question. So what about VP30? What is the whole big deal with VP30? Why are people so excited about it? Why am I, again, concentrating on this a lot? Well, what happens in these filoviruses, which again is different from the paramyxos and the rhabdos, is that many of the messenger RNAs, not just the nucleocapsid protein, have these really strong secondary structures that form in the messenger RNA at the 5' end of them. And this hairpin seems to block transcription to some extent, but also translation. So you don't actually make this messenger RNA, and much more importantly, if you think about how you make the messenger RNAs, it's the L protein, which is coming along and hits one of these sequences and goes, oh, okay, we're going to do this. But these secondary structures, fine, it's an opposite strand, but they're always still going to be base pairing with each other. So this could be positive strand, it could be negative strand. You're always going to have these really strong secondary structures. VP30 seems to denature this and pull them apart. These sequences are basically identical in each of the messenger RNAs and the rest of the genome. VP30 allows this to fall apart and get nicely transcribed. Evolutionarily, how this developed, I have absolutely no idea. It's a really interesting question, and I don't have a good idea. If you guys have some, let me know. But for filoviruses, it's really critical that you have this VP30. Mentioned before, where do you do these experiments? This is actually one of those labs. This is in Galveston, Texas. This is their BSL-4 lab at the University of Texas Medical Branch. Kind of looks like a pretty normal building. Unfortunately, none of these windows actually go to the outside. And it's also built in such a way, you may notice that there's a couple of windows right here in the middle where it looks like there's absolutely no window whatsoever. That's because this BSL-4 lab is actually built inside a BSL-3 lab. So the outside of the building is BSL-3, which actually is also not allowed to have any windows. And then inside that is the BSL-4 facility. Looks like, again, a pretty normal office building. You go through here, and then you get into the security aspects of things. And so it becomes pretty obvious. These bollards here as well. One of the big issues that I was really kind of concerned about, anybody remember what happens in places like Galveston and the Gulf Coast? Yes, you have oil spills, but that's not a big deal. Hurricanes. So my thought was, well, why the heck are you going to build this high security? You've got Ebola, a live virus in here, right in the path of a hurricane.
Is it on a hill at least?
No, actually, this is pretty much at sea level. Have you ever been to Galveston? Hill just doesn't part of the vocabulary there. It's really, really flat. So they had a hurricane, which was Ivan, I think, was the one that actually directly hit here. Katrina, of course, was an issue there too. They flooded the whole basement of this building, completely flooded. The BSL lab was up and running a week later because they designed it for hurricanes. And you'll also notice it's up. It's not all the way down at the bottom. And I actually met the guys who were involved in the design of this, and they said they actually preferred hurricanes to something like a tornado or an earthquake because you can see hurricanes coming. Which at first it's like, oh, that's a really bizarre idea, but it makes sense. They've got a whole thing was designed so if the hurricane comes, you shut everything down, move everything away, lock it all up, and then a week later they're back up and running. Tornadoes also have worse winds, and they're talking about building a new biosecurity lab where? Anybody know? Kansas, Manhattan, Kansas. What do they have in Manhattan, Kansas? Take me back to Kansas. Go home. Click the heels together. So yeah, there's some really interesting design kind of issues to do that. So this is where these kind of experiments actually get done, and so. This is how you go about making those kinds of mutations in your genome. And this is a lot like what we talked about before when we talked about the different coronaviruses. Making the individual pieces and then sticking them back together. So you have your DNA copy of your RNA genome. This is the complementary DNA here for your Ebola virus genome, together with your T7 promoter and T7 terminator. Here, one of the things that's a little bit different, there's a ribozyme, which is encoded in this particular part of the genome. Why? Any idea why you want to have a ribozyme there? What would that ribozyme be doing, potentially? So, do you want to have a T7 promoter and a T7 terminator at the ends of your RNA if you're going to try and make it virus? No. So what these ribozymes do is actually clip the ends off. They're basically like self-splicing introns. So they cut themselves off. Really smart idea in how you do this. So you can make the RNA here really nicely. But this is not like you have in coronaviruses. What about coronaviruses? They're nice positive strands. All you need to do is take that RNA and put it into a cell, right? But to make an infectious negative strand RNA virus, what do you need to do? It needs the polymerase, it needs all of the nucleocapsid proteins, etc. So you have to be making this in a cell that also makes all of the appropriate proteins that you need. So the L protein, the NP protein, the VP30 protein, etc. And then you can make this nucleocapsid, which will make the viral messenger RNAs. So it's a pretty complicated process to go through making any one of these recombinant viruses. And if you think about, and we'll take a picture, take a look just a second at how you actually have to work in some of these BSL-4 labs. You've probably seen the whole suit thing. It's really challenging to do these kinds of experiments. So now we'll switch bases and talk about, oh yes, the wonderful diseases. Marburg virus. Marburg virus? Why Marburg? Anybody been to Marburg? Me? It's this wonderful little place in central Germany. Has a nice castle. This is actually the castle in Marburg right here. Identified in Marburg in the 1960s because a couple of people who were making vaccines, not to these particular viruses, were working with monkey cells, and they all came down with this really bizarre disease. And then, later on found in Belgrade as well. Eventually, people figured out that the reason they were coming down with this disease was because the monkey cells, which had been imported from Africa, which were actually infected with these particular viruses. So Marburg is actually rather confusing. Again, it's really a tropical disease, particularly in Africa. So Ebola is the other favorite one, and again, we'll talk a lot more about this. This is just up to 2005. 500 cases of Ebola in the world up to 2005. The first one of the filoviruses was discovered in the 1960s. So 45 years, 500 cases. Not very many. These were the different filovirus outbreaks that had happened in Africa. The original outbreaks of the African filoviruses were Ebola, and Ebola is named after the Ebola River here in the Democratic Republic of Congo. Turns out that there's a Zaire. Now, why Zaire? Because that's the name that the Democratic Republic of Congo used to have. And in the Sudan, more or less simultaneously with each other. And then sporadically, every 5, 10 years or so. Interestingly enough, there was a case of Ebola virus here. Anyone know what that state is? No, right here. This one, where the arrow is. Honey, that ain't Virginia. So, African geography. Who knew about African geography? I was going to ask that question here. Should have had that for my clicker question. So this one over here, 94, Ebola outbreak. Côte d'Ivoire, exactly. Ivory Coast. So not completely unheard of in West Africa until last year, but in 20 years previously. And that's probably one of the major reasons why this outbreak has been so nasty. This one, on the other hand, yes, is Virginia. Reston, Virginia. Why Reston, Virginia? Monkey house, exactly. Why a monkey house? Well, because all the monkeys died, but why would there be a monkey house in Reston, Virginia? For research, why? Okay, what's this place here? Bethesda. What's at the Bethesda? It's the NIH. So all the monkey facility for the NIH is in Reston. So that's where they had a bunch of monkeys die. And it wasn't all of them, but quite a few of them. And it turns out from a filovirus disease. And then they said, okay, where do these monkeys come from? Come from the Philippines, not from sub-Saharan Africa, which is where they'd found all of these filovirus diseases before. And I've now gone back to the Philippines, and they've found a number of monkeys that are infected with this disease, probably also some of the reservoir species, which, again, not unlike what we've heard before for many of these other viruses, seems to be bats. So probably the bats there have this Reston Ebola virus. Why is Ebola in Virginia so good? Well, not a normal place for it to be. Okay, yes. Not tropical, although sometimes in summer Virginia does feel kind of tropical. But no, the main thing here is it doesn't seem to cause disease in humans. Non-human primates, yes. Humans, no. And so it's a really nice model system for studying these filovirus and trying to understand what's going on there. Of course, it doesn't say that much about human disease, but in terms of the molecular biology, molecular virology, these filoviruses, Reston is a really, really good isolate to work with. Okay, why do we care so much about it? Well, again, hopefully now with this more recent outbreak, it's become a little bit obvious. Messy, although it's only messy in some cases, which is the hemorrhagic fever. The main reason that people seem to get really, really sick and die doesn't, again, seem to have to do that much with the virus infection itself. It's the immune response to it, and particularly massive numbers of macrophages which are being produced. It's clearly, as we've talked about with many of the other cases, a zoonotic disease, which means it comes from cute little animals, well, depending on how cute you think bats are. One of the big problems that people have had with all these outbreaks of these filovirus diseases is very often it's medical personnel which are coming down with these diseases and getting really, really sick and dying. Also, very close family, and just the case between these two means that you probably have to have really pretty close contact to have any kind of transmission, and we know that really well now for Ebola and the West African case as well. It's also clearly immune suppressive, and again, the problem is here probably has to do much more with your response to it rather than anything else. Probably the main reason that we care about this has to do with the media, because at least until 2013, there were 3,000 cases ever of filovirus disease, relative to the other things we've been talking about. What about, well, say dengue? Tens of thousands a year of dengue. Measles, probably 1 million or so per year. 3,000. Yeah?
Do you think that these filoviruses have existed in animals for a really long time, or are these like is it just new that they're infecting humans and that we've noticed them?
So the question is, how old are filoviruses, which I think is a wonderful question. And the answer is we don't know, we being not just me. And people are trying to figure that out right now. And when we get towards the end of the lecture today, we'll talk a little bit about that, how they're trying to go about figuring this out. Clearly in humans, well, not clearly, but probably in humans, it's a relatively recent thing. But how long these kinds of viruses have been circulating in the animal populations, nobody really knows. And it certainly seems that in the few cases that we've looked at, the bats, there doesn't seem to be any kind of disease. And so that will be a nice indication of how they actually could be maintained for much longer periods of time. Yeah, so very few of these. Again, if you don't listen to TWiV, they're actually going to change it to This Week in Ebola Virology, because they had a huge number of, this is, again, the podcast for this. I love this title here, No Reston for the Weary. And so these are the numbers they had here. Since 1967, 2,800 reported diseases, less than 2,000 deaths, only 50 known introductions into humans. Turns out it's pretty wimpy virion, heat, detergent, light, you can get rid of. This actually means it's kind of tough to get viruses, and that's one of the reasons why answering your question has been a little tough, because you go and you collect bat guano. Anyone want to go into a bat cave in Ebola or Marburg fever areas and collect guano? Not something I would really want to. You might dress up like this. This is, again, plug for the film on Friday. But, yeah, I don't know of any people at the CDC who look quite like Dustin Hoffman. Update in 2012. This is, in fact, my lecture slide from 2012. There was 1 case in 2011, 7 cases in Uganda in 2012, 24 cases also in Uganda, separate outbreaks, and 77 cases in the Congo. So again, relatively small numbers here. Marburg, on the other hand, again, not endemic to Germany, but in Uganda, 2012, 15 confirmed, 15 possible deaths. 1 tourist has died in 2012 from any of these filovirus diseases. So if you consider the amount of media outbreak, et cetera, that's happened here, I think it's more of a media outbreak than it is a disease outbreak. Wonderful website here at the CDC that we'll not take a look at. So when I was putting together my lecture today, I noticed my lecture from last year, 2014, 2nd of May, that there was a new outbreak of Ebola. 234 cases in Liberia and Guinea, 146 deaths, about a 62% fatality rate, which is pretty normal for diagnosed filovirus infections. Unfortunately, lots of healthcare workers. This is, again, the 25 healthcare workers and 16 of those who had died. So, again, a pretty similar kind of fatality rate. This is one of the CDC pictures that they had there early on in the outbreak. What would that look like? Again, from April of 2014, outbreak was confined to this region of Guinea and a little bit of Liberia, and at that point, no outbreaks in Sierra Leone. So what does this place actually look like? This is Kissidougou, which is one of the provinces in Guinea, very close to where the outbreak seemed to have happened. This is the mall in Kissidougou. I don't know how many of you have been in sub-Saharan Africa or particularly in West Africa. I've been in West Africa, I've been in East Africa. This is the big city and the market in the big city. This is where probably, let's see if I hit the lights here a little bit more, the first case of this particular outbreak happened. This is Meliandou in Guinea. This is really typical for the way many, many people live in that part of the world. This is the main road right here. So clearly hard to get through here, but a lot of people have motorbikes. Motorbikes, mopeds, etc. So either walking or on motorbikes, it's really hard to get to in general. Think about hospitals, medical relief care, etc. But people can certainly move around. Where is this? Again, it's in Guinea. But these original outbreaks are right here at the border between Liberia, Sierra Leone, and Guinea. Just reading a New York Times article, and I'll give you the link in just a second here. They say that basically this was sort of the worst place to have one of these diseases arise for a number of reasons. These are some of the poorest countries on Earth, which hopefully you get that idea from looking at that last slide there. A lot of these borders were drawn up by other people, and cross-border transport is extremely common. Until very recently, there were civil wars in a lot of these places, so massive numbers of refugees, particularly in Kissidougou, again, this big city right here that I just showed you the mall from. And this is the site, literally, of that one village. People have looked at the outbreak statistics from there, probably case 1 right here, end of December of 2013, and then increasing here into March and April of 2014. And remember, May, that was when I had my lecture about this last year. It looked as if numbers were actually going down here. This is exactly what had happened in all of the other Ebola and Marburg outbreaks. Everyone thought, and even me, when I was giving my lecture then, this is probably it. It was probably over after a couple hundred cases. It wasn't likely to be continuing. So we clearly did mess up about this. And so this, again, there's a nice long, actually, New York Times article about this. But we messed up for a couple of reasons. One, having to do with the really amazing cases of poverty which are there. In terms of following up on any of these filovirus cases, as I mentioned before, really close contact, usually actually physical contact between whoever has the disease and whoever is going to get infected by it. And the sicker you are, usually the more virus you're actually producing. So the sickest people, direct contact. Unfortunately, that's usually the medical providers who are doing that kind of thing. The other thing has to do with the culture of that region, which is at burial, when somebody dies, there's usually a lot of direct contact. Washing the body, kissing the corpse, physically handling this. There was, in fact, one case that I read about, but I wouldn't go all the details here. I can give you the link if you're interested, where they had one guy who had died, and then 18 people at a funeral who had actually literally either washed the body or kissed it. And this was at the beginning of 2015. So it'd be out there already for a year. And still people were doing exactly the same kinds of things. Fortunately, that particular outbreak was confined relatively quickly, but it was already 100 cases, basically, before that happened. So showed you here what it did look like. Oops, here we are. And now, almost exactly a year later, this is what the case looks like. So between 500 and 4,000 cases, those are the really dark ones here, has spread from Guinea, Sierra Leone, Liberia. One of the major problems here is that we've gone from this very remote, mostly really jungle areas, to the main centers of population. So the capital of Guinea is over here, Conakry, capital of Sierra Leone right here, and capital of Liberia here. One of the things that has really helped now, this is pretty nasty, we'll show the numbers in just a second, is really now going to this kind of control systems. You remember I showed you that picture of the CDC scientists pipetting with gloves? Now this is the appropriate way of doing this. And setting things up properly. Maybe a little hard to see here. I'll turn the lights down again. This is another picture from that New York Times article. Hopefully you can see here. This is the dressing tent where people are being dressed and undressed by somebody else, again, to avoid this direct physical contact. This particular Ebola treatment site is through Médecins Sans Frontières, which was really sort of the major group that A, said, yes, we screwed up, and B, got a huge number of boots on the ground, and this is in fact their publication here about dealing with that. Because of these kinds of measures, if you look at recent cases, the map looks a heck of a lot better. So if you look here, this is now immediate cases, 0 days, back up to greater than 63 days. Most of these new cases are here in the major population centers of Sierra Leone and Guinea. But if you look back here, where the original outbreak was, 80 days, 130 days, 180 days, and even in Liberia, 34 days. And these are the last updates here. This is the 23rd of April, 26th of April, 26th of April. So if we look at the total numbers as of the 1st of May, about 26,000 cases and almost 11,000 deaths. So you remember up until a little over a year ago, it was on the order of 3,000. So one outbreak now on the order of 11,000. So definitely nasty, but on its way down. Whether it's on its way down because of infection control is probably the case, or it could just be one of these self-limiting things, which we've seen with all these other outbreaks of the disease as well. But that's nowhere near as important as Ebola in the US, right? So 11,000 deaths, 1 in the US. And that 1 was misdiagnosed. Came into the hospital in Texas, was sick, got sent home, got really, really sick, came back, infected 2 other people because they didn't have the appropriate precautions, probably. Again, that's still a bit up in the air. And then there was another case of a doctor who'd been volunteering who came back to the US and turned out to be Ebola positive, who admitted himself into the hospital. So did exactly the right thing, which is there. I love this graphic, which I found putting together yesterday. Americans concerned that they or their family will contract Ebola. Now, this was admittedly August of last year. 51 million people. How many had actually had contact, have contracted Ebola, at least in the US? None. So this is, again, I don't know why we consume these kinds of media, but yeah, fear definitely is outstripping reality. So what are early symptoms of Ebola? This is all stuff from the CDC website. Symptoms: fever, headache, fatigue, diarrhea, vomiting, stomach pain, unexplained bleeding or bruising, muscle pain. Okay, these are all pretty general symptoms. And that is, in fact, also one of the reasons that diagnosis was difficult early on, and really showing that whatever you've died from is not malaria, which causes almost all of exactly the same things here and kills that number of people in Western Africa every year, or Lassa fever, which is also there, and it's one of those Bunya viruses and arena viruses we're not going to talk about, also causes a lot of these same kinds of symptoms as well. So you do have to be very careful about the diagnosis here, and at least to start with, the diagnosis for the first cases was done in Paris. So you had to get the samples from the back of Guinea all the way to Paris to actually get some kind of diagnosis here. So top things you need to know about Ebola. Your dog or cat is not spreading Ebola. People got that idea. Food and drinks from West Africa are safe to eat and drink. Mosquitoes are the deadliest animals in the world. Yes. Dengue, well, the arena virus as well, but they don't carry Ebola. No reports of mosquitoes. Your family's co-workers returning from countries don't pose a danger to you and your family unless they're really, really sick, in which case they won't be at work. And they won't be, hopefully, interacting with you. Household bleach and other disinfectants kill Ebola. Yes, they do. If you're feeling sick, it's not Ebola. Flu, there is a vaccine for flu. Is there an approved vaccine for Ebola? No. Talking to my GP when this was all happening, she had said more people coming into her office asking for an Ebola vaccine than a flu vaccine. Okay, Ebola is not airborne. Those of you who've been listening to TWiV know that they really get into this. Is not affecting the safety of airline travel. How many cases of Ebola transmission on a plane? 0. Absolutely none. Spread from one person to another once you have symptoms. Although there was a report just recently, and one of you sent me an email about this. Semen transmission. That's a really interesting question, whether you can have semen transmission. There have actually been 2 reported cases of transmission, one of Marburg and one of Ebola, of about 15,000 now, that have been reported to be transmitted through semen. And in fact, one case after someone had recovered. That was the recent one, which was just published. Still rather controversial whether this is the case. Certainly not a major transmission mechanism, but something that might be happening there. Still, again, very open question about that. And no, you can't get Ebola from a handshake or a hug. So again, it's amazing to me that this even had to be published. But it's there. Next time you're in the biology office, check out the flyer. So if, however, you do happen to be in West Africa, you are a healthcare worker who is there, how can we go about dealing with this? So there are a number of potential vaccines. Not surprisingly, the GP has been the main thing that people are looking at in terms of trying to use this. Recombinant viruses, which we mentioned last time. VSV, again, not terribly surprising because it's really similar molecularly to any of these filoviruses. What they did is they took the GP protein and expressed it on VSV, just basically swapped out the G protein for the GP protein. Seems to be working reasonably well. People are also thinking about using RNA interference, double-stranded RNA, to try and deal with this. There is a vaccine trial going on right now in West Africa. This particular one is in Sierra Leone. Started almost exactly a month ago, a little less than that. So really interesting clinical trial. So I was reading about this last night. So it's an unblinded trial. Okay, well, what does that mean about your data that you get back? No. But is it morally reasonable to have a vaccine that you think works, and you're testing at people who might be coming down with the disease. So in this case, you know if you're getting a vaccine, and everybody in the trial gets a vaccine. The way that you actually get any kind of data from this, because you need to see if somebody is actually, the vaccine's doing anything, because if everybody's got the vaccine, how do you know if it's a difference between having the vaccine or not having the vaccine, is that some people are getting it 6 months later, which again, at that point, the disease may be gone. But that's the idea. That's how they're doing this trial, which is a really kind of a new way of doing clinical trials. Usually you've got placebo and your actual effective thing here. At this point, everyone's going to get the vaccine. There's going to be slightly different. And they get to choose, actually, which they want to have one now or later, because we also don't know completely about the safety of this vaccine. So you can take your pick. And you also can say, no, I don't want to participate in this at all. Almost all the people, they tried to sign out 6,000 of them, are medical professionals, people who are dealing with this on a day-to-day basis. And there clearly are still cases of Ebola that are circulating in West Africa right now. And they're using this recombinant VZSV with the glycoprotein on it. Yeah?
This doesn't seem to be enough people to do a phase 3.
So the question is here, it doesn't seem to be enough people to do a phase 3, 6,000 people. And the answer to that is no, it is a pretty low number. But the only way to actually have an efficacy is if you have an outbreak. Well, if you have an outbreak, how many people are you actually going to have? And so this is, in fact, probably the best opportunity that we have to actually test any of these vaccines. But it is a really open question. Now, these things work really well, and they must have worked really well in non-human primates, because otherwise they wouldn't even be here, where you can actually do some of these controlled experiments. But out in the actual real-life world, do you want to take an Ebola vaccine that hasn't been tested on people? Okay, but do you want to come down with Ebola? These are not easy questions to answer. They're really, really tough to try and answer. So you may have a nasty disease here that has nothing to do with Ebola, but you may be protected from Ebola. There's another vaccine, again, that was literally just published about a week ago in Science. This is that deletion of the GP30. You remember the GP30 is absolutely critical for getting your transcription, or at least making messenger RNAs, getting rid of those hairpin structures that are at the 5' end of each of these messenger RNAs. You can make a virus that is lacking that protein if the cells are making the proteins that it needs to make the actual virion. And so basically what you have is you have this deleted version, which again you've made, in this case it was in Hamilton, Montana, the deletion of that VP30 gene. The cells that you grow your viruses on have the VP30 protein in them because you couldn't make the whole actual virion without it. But then you purify the virions away from that. So that virion has a genome that's packaged with VP30 because that came from the cell it was growing on. But the genome has no VP30 in it. So it will infect. It infects perfectly normally because it's got the regular GP proteins on it, everything else. And then it tries to make its messenger RNAs. It can't. Gives really good immune responses and has worked really well so far in non-human primates. But unfortunately, also with non-human primates, yes, it's hard to get 6,000 people for a clinical trial. Most of these primate studies are tens because it costs tens of thousands of dollars for each of these primate studies. So they're incredibly expensive. So there are vaccines in the pipeline. How good they are, it's not even clear whether we're really going to know what's going on there. So what about Ebola? Where is it coming from? Well, probably, again, is circulating in some reservoir species, probably bats, but again, there's an open question about whether that's the case. Human infection, not clear whether it's directly from the bats or some other kind of non-human primate bushmeat. In this particular outbreak, they say there was a tree and there were bats in the tree that the poor little kid who was patient 0 was playing underneath. Really hard to tell what's going on there. But the idea here is that, again, most of the virus is here and occasionally transfers to humans. And then in the case of Ebola virus disease, if the humans are really sick, And usually in the really close families, this is where you're getting transmission that's actually taking place. So what are we doing about this? One of the main things that the CDC is doing is trying to figure out that question that you were asking right at the beginning. Where are these viruses most of the time? So what they've been doing is literally going into some of these bat caves, and again, this is not a job I would want to have, and screening bats for really any kind of viruses, but filoviruses as well. And the way they're doing that is just collecting the bats, sequencing everything they can find in them, bat DNA, virus RNA, etc. And hopefully in that process, figuring out where some of these things have come from and what's the diversity in terms of the sequences, which you actually have there. So that's what's happening there right now. To finish off, I have a short video from my friends at the Canadian Broadcasting Corporation on what some of these BSL-4 labs actually look like.